Hello, my name is Grace McLaughlin and I'm a Master of Infectious Diseases student at the University of Western Australia. This study, Bacteria in Human Breast Milk, do staphylococcal species preferentially metabolize lactose, investigates the relationship between Staphylococcus aureus and the lactose concentrations in human breast milk. Human breast milk is important for the proper growth and development of infants and is both a source of nutrition and a source of microbiota. Lactose is the main carbohydrate in breast milk, and studies have shown that low lactose breast milk contributes to poor growth and increased risk of disease and undesired early weaning. This study follows on from our previous study that examined the relationship between the human breast milk metabolome and microbiome of African mothers. A subset of breastfeeding mothers was identified as having low lactose concentrations in their breast milk. Our previous study showed a clear association between staphylococcal predominated microbiota and low lactose concentrations as seen in Figure 1. The findings suggest that dysbiotic human breast milk microbiomes result in increased lactose fermentation, leading to low lactose concentrations and poor lactational outcomes. A study by Richardson et al. Published in 2018 found that Staphylococcus aureus isolated from bovine milk exhibited enhanced lactose utilization. Therefore, we hypothesized that Staphylococcus aureus found in low lactose human breast milk will have a greater ability to utilize lactose compared to the strains found in human breast milk of normal lactose concentrations. To test this hypothesis, we first enriched the human breast milk in various nutrient broths, as seen in Figure 2. The broths were then plated onto selective and non-selective agars to maximize the likelihood of isolating all staphylococcal species present. Staphylococcus aureus was then isolated and identified through gram staining, catalase, latex agglutination, and DNase tests. From this, we saw a greater proportion of low lactose human breast milk samples containing Staphylococcus aureus compared with human breast milk with normal lactose concentrations. We then screened the Staphylococcus aureus isolates for lactose fermentation using various concentrations of lactose as seen in Figure 3. From this, we determined the best concentration for Staphylococcus aureus growth was 100 millimoles. However, we also decided to use 300 millimolars in our lactose fermentation assay, as this is close to the natural lactose concentration in human breast milk. We are now currently running a lactose fermentation assay using the method published in the Richardson et al. 2018 paper to assess the isolate's ability to utilize lactose. Lactose fermentation will be measured by the pH change in the supernatant. And if Staphylococcus aureus isolates from low lactose human breast milk are found to have an increased ability to utilize lactose, then identification of these strains may allow for interventions to improve breastfeeding outcomes in the future. Thank you for listening to my presentation.